We start with a keynote lecture by Professor Karin Allenspach. She's a professor at the University of Georgia and a clinician scientist well known for her work on intestinal organoids and to study intestinal disease and immune processes. Professor Allenspach is not with us live today, but she kindly recorded her presentation for us. So, Karin, thank you for being here, and we can send the first video with Karin.
Hi, everyone. This is Karin Allenspach speaking. Thank you very much for inviting me to the IBD Day in Nantes. Unfortunately, I'm unable to be there in person, but I'm happy to provide this video of the presentation, and hopefully it will spark some good discussion. So we're going to be talking about canine intestinal organoids, current uses and pitfalls. So just as an introduction, one of the problems that's been discussed recently, both in Europe as well as in the US, is one of the bigger problems that we have with regards to drug development, right? And it's really about two big problems. One is efficacy testing, as in predicting whether a drug will actually work in humans and what kind of model systems we have to investigate that before we move into clinical trials in humans. And the other one is safety, which, obviously is also really important because you want to make sure that the drug is safe before you put it into a human. But overall, the whole process is actually pretty inefficient. Only about 5% or less of all of the drugs that are being, developed in biopharmaceutical industry actually make it to market eventually. And per drug, that means that there's about $2.7 billion being spent, and it takes anything from 12 to 15 years or even longer to actually get a drug to market. So, again, that means that we're not doing that well with regards to modeling accurately what we actually want to predict with regards to efficacy and safety. And so ADME is one of these things that as a pharmaceutical industry is really important because it stands for absorption, distribution, metabolism, and excretion of drugs, and is one of the things that you want to pay close attention to and try and get some preclinical information before you actually try the drug in humans. And then preclinical drug testing, in addition, obviously also is about more than just, pharmacology and pharmacokinetics and pharmacodynamics, but also about safety and efficacy. So what I think people have been talking about recently, particularly with replacing some of the in vivo models, particularly mouse models, with potentially organoids, is can we use intestinal organoids, for example, for toxicity testing or for permeability absorption testing? Because the problem really is that 2D cell lines don't predict very well, how much a drug is absorbed or how much it is permeable to a drug. So there is definitely some things that have been happening, particularly in the human space, where 3D organoids or even microfluidics are, beginning to replace some of these healthy in vivo modelings. And then the other thing is, can we do better than in vivo drug screening in healthy dogs? Because that is also still one of the steps that's being done in pharmaceutical industry before moving a drug into humans. It's usually what they call a large animal model, in this case, a dog, where they check for all of these things beforehand. And then the second thing is really about efficacy testing, where, a lot of the problems really lie because it's really difficult with particularly mouse models with induced disease to model whether a drug will actually work, because the naturally occurring spontaneous disease in humans is usually quite far from what we are modeling in mice. And so there, we also probably have better models, as we know. You could actually take organs from dogs that have analogous diseases to people and develop 3D organoids from them, and then actually use canine clinical trials before you move a drug into humans. And those are things that we're going to be discussing today. So going forward, one of the things that's really come up recently, particularly in the US, that, there's a lot of talk about not having to do a lot of mouse animal testing anymore. And in fact, the FDA Modernization Act has a clause in there that basically lets pharmaceutical industry decide what kind of preclinical testing they want to present to the FDA in order to get approval for a drug on clinical trial in humans. And so if they feel that, for example, a mouse model is not going to be providing helpful information, they are allowed to actually use something else. Probably, obviously, still is up to the FDA to then believe that and see whether that satisfies them. But it is sort of a step in the right direction. So the technology that we're talking about here is the so-called adult stem cell derived organoid technology, which has been around for about 10, 15 years now, and really started off with intestinal organoids in humans. And I'm going to show you a little bit about that. So what are organoids? So organoids were actually developed on the basis of an in vitro problem that we have, which is that the in vitro modeling of primary epithelial cells from patients, whether that's veterinary patients like dogs or human patients, is actually really difficult to do. And that's because primary epithelial cells and their stem cells do not survive for longer than a few days in culture. And that's because once they differentiate, they have a finite amount of time until they undergo apoptosis. So if you think about the intestine, usually the renewal is about every 3 to 5 days. And so in that time span, that intestinal stem cell has differentiated itself to a terminal differentiation state, for example, an absorptive enterocyte, and will then undergo anoikis or apoptosis, and it will die. And so to overcome that, this technology of three-dimensional organoid culture has been developed about, as I said, 15 years ago. And what it does is basically it gives the stem cells a little bit of a structure surrounding them with something called matrigel, which basically gives it a little bit of a structure so that it can grow three-dimensionally. And then you also give it growth factors, and they're specific for the animal, so for the species, as well as for the organ that you're trying to culture three-dimensionally. And then what happens is that that stem cell will basically remember what it was doing in vivo in that animal or in that patient, and it will grow up and differentiate all of the cells that you had originally physiologically in that animal at that time. So it represents the genetic as well as epigenetic background of the tissue of origin, and it lets you then basically culture it forever, which is a really huge improvement because with 2D cells, you only have one clone, so just one cell, and it's usually not representative of anything that you see in vivo. So how can we use this? And I've provided an example here that I want to show you a little bit of data on. So this is about intestinal permeability and absorption that you can measure using organoids. So again, why would you do that? Because 2D cell lines are really inaccurate because they only have one clone. And if you're going to look at this anyway in vivo in dogs, I think it would be really nice to be able to cut down on the numbers of dogs used if we can actually use 3D organoids first. So what we've done is come up with intestinal organoids for dogs for the first time. And this paper came out in 2019 in BMC Biology. And what you can see here is that these stem cells grow into sort of cyst-like structures within about a day or two. Then they have these little out spurts that are the crypt-like domains. And then that becomes more and more. And within about 7 days, they have grown big enough so that they need to be passaged, as in broken down again. And then you do that every week. And if you wanted to, you could continue indefinitely doing that. So we can do that with intestinal endoscopic biopsies taken from healthy dogs as well as diseased dogs. And what you have to do is just be deep enough so that you get crypt cells, which of course you would try to get anyway if you're trying to get diagnostic biopsies as well. But these are going to be the cells that you need in order to actually have a growth of an organoid in your in vitro culture. So how do you culture organoids? This is kind of a schematic that I made to try and explain why organoids could be better to look at something like permeability, absorption, and metabolism. And it's because if you have, for example, KCO2 cell monolayers, which are the standard of how this is being done for drug testing, you have one particular cell type, right? Which is the clone of the KCO2 cells. That was actually originally developed from a colon carcinoma cell line. And so it's it basically only gives you some idea about paracellular transport, and that will be it, because everything else will be different from what you're testing here and what actually happens in vivo. So in vivo, of course, you have a lot of different cell types. So you have absorptive cells, you have enteroendocrine cells, you have goblet cells. So all sorts of things happening, and what you can look at, for example, is metabolism of drugs, like the breakdown of drugs within cells, the efflux by certain transport proteins like P-glycoprotein, for example. Again, the paracellular transport, but also the transcellular transport, as in what kind of transporters will actually shuffle drugs from one side to the other. So that's the idea of using organoids and being better with modeling what happens in vivo. So this comes from the paper that I just mentioned from 2019, where we did this in organoid cultures from dogs. And this is just to show with using transmission electron microscopy how much these cells mature over about a week. And what you can see here is that on day 3, you still have a very big sort of intercellular gap here with some desmosomes building, but there's still quite a bit of a gap here between the cells. And then by day 6, you see a lot more microvilli coming up, and the gap seems a little bit smaller. And then by day 9, they're completely matured. They also have mitochondria within the cells. They have not just microvilli, but they also have mucus production, and they have fully formed desmosomes and adherence junctions that you can see here. So it's a fully matured, differentiated cell that basically you can keep growing forever if you wanted. We also were looking at expression of specific markers that would show us that we have differentiation of all different cell types. And so we were looking, for example, at SOX9, which are intestinal stem cells, ALP1, of course, for enteroabsorptive cells, and then also some markers that show that some of these cells have the function of Paneth cells, even if they don't look like it under the microscope as in humans. Well, the next step is, of course, if we want to do any sort of permeability or absorption measurements, you need to actually open these intestinal organoids up again, because what they're doing now is actually having their apical membrane looking towards the inside of the cyst or that little organoid. So that's inconvenient because you kind of want to access that apical side, because you want to, model how a drug that's absorbed, orally given is actually being absorbed through the epithelium. And so in order to do that, you can take the organoids and culture them back in a 2D model, which sounds a bit counterintuitive, but, again, it's necessary in order to make that accessible, that apical membrane. And in order to do that, you basically culture them on trans wells. So that's kind of an insert within a well of the culture. And you have media on the one side as well as on the other side. And once you do that, and once you have a confluence of all of your organoids, you basically have a monolayer of the cell as if it was in vivo in the intestine, but that will only last for about 4 to 5 days after they're confluent, and that's the time span that you have to do any experiments. After that, they will be gone, and it's similar to any sort of primary cell culture again. So there's some more pictures to this. So what you can see is, again, sort of them growing up from day 1 to day 5. And then when you put them on the trans well, you wait until you have this confluent layer, which basically then means that you have your starting point from when you can do your experiments. And this is another nice picture from that publication that we had in PLOS One, where this is zonulin and this is ecadherin that you can show that is nicely expressed in these monolayers. And then again, some transmission electron microscopy showing desmosomes. And we were also measuring transepithelial electrical resistance. And this is important because the monolayer, unless it is completely confluent, will of course not really show you any, relevant permeability or absorption. It will just flow through if there's holes in your monolayer. So in order to be sure that you have a confluent monolayer, you can actually measure that transepithelial resistance. And once it hits a peak, it will basically only go down very slowly over about 4 to 5 days. And this is the window that you have to do your experiments. So to show you a few results, this is a paper that we've had out, I think, in 2023. So this is looking at the in vitro model system of canine intestinal organoids to model intestinal permeability of therapeutic drugs. And this was a collaboration with the US FDA, where we chose drugs that they have a lot of background knowledge on and that they have a lot of knowledge and data on because they were used a lot on KCO2 cells as well. So what we were doing here is basically KCO2 cells versus intestinal organoids in humans versus intestinal organoids in canines to show what the differences are in absorption of these drugs. And so what you do is basically give it on the apical side, and then you measure it in the basal compartment over time to see how much of it is being absorbed. And we were looking at propranolol, metoprolol at 2 different concentrations, and then atenolol as well. And what we could see basically is that there are some differences between drugs, particularly between propranolol and metoprolol. If you look at either canine or human systems, which, is relevant because that is one of the things that you also obviously shouldn't forget about. I think as a veterinarian, that makes a lot of sense. But, for human drug specialists, actually, just measuring something in dogs does not necessarily mean that you have the same sort of permeability and absorption going on in humans as well. What was interesting to see as well is that with this organoid system, in the human system, atenolol was actually absorbed really nicely, but it was not absorbed at all through the canine intestinal organoid. And that is interesting, right? Because we know that we can give atenolol and that it is actually absorbed in dogs. And so, I'm showing you this as an example of sometimes even with organoids, we're not quite there yet. And there's probably more things that we need to consider and take into account in order to make the system perfect or as close to perfect as possible. Because otherwise, again, we can be wrong with the predictions that we're making. So moving on to the next example, which is about efficacy testing. Why do we need this? Because mice with induced disease don't always accurately predict efficacy of drugs in humans or in dogs, speaking as veterinarians. And so 3D organoids derived from diseased human organs, would be preferable, of course, if we're looking, to actually get a drug to the human market and eventually test on humans. But the problem with this has really been that organoids from organs from humans that have disease are difficult to obtain, and that's for ethical reasons. And so there's really not a lot of larger bioarchives that have become available. If we use 3D organoids of intestine of diseased organs, for example, in dogs first, I think, again, we can use this to narrow down on which drugs to test in preclinical trials using diseased dogs and then go into humans. So that's kind of the idea behind this. So one of the things that I want to show you here is that we have over the last 4 or 5 years put quite a bit of effort and time into the development of an intestinal inflammation assay that is repeatable and that we can test drugs on for canine intestinal organoids. So what we've done is basically developed a test with a couple organoid lines from dogs, from healthy dogs, and have basically shown that we can repeatedly get the same results. And this is really looking just basic at cytotoxicity using DMSO. The higher you go with that, the more damage you get, basically. And so the more cytotoxicity you get. And that's actually also true over time. So, the highest amount of cytotoxicity that you can see here is at about 6 hours after giving DMSO concentrations. And then it's different if you actually expose it from the apical or from the basal side, right? So if a cytotoxin is coming from the blood stream, basically, then you have to give more of it and for longer to get similar effects. So those are the things that we just started with to sort of explore the assay system and see if we get it consistently to run. And then what we did was we used canine TNF to stimulate the healthy intestinal organoids from dogs and see if we can stimulate IL-8 in the supernatant. And the idea was really to have something that is a consistent model of intestinal inflammation that has been exposed to TNF and then produces IL-8 as a response, that we can then use for testing anti-inflammatory molecules. So, if any kind of a biopharmaceutical industry for animal health comes along and says, we really want to know whether our agent or our compound has an anti-inflammatory activity on intestine in the dog, then this is a really good first step because it gives you an idea of whether your compound is able to decrease that IL-8 output over time. So this is just without any additional drugs. What we consistently see is if you give TNF in the apical side or in the basal side, you can see it comes up about 24 to 48 hours later, and you get a nice consistent response with IL-8 being produced by the epithelial cells. This is the other thing that we can see here as well. So when you look at TEER values, again, is transepithelial electrical resistance. So it takes a little bit of time until that comes up to a level where it stays a little bit more consistently up. And then, if you give TNF at this time point here, 10 days after seeding, then you can see that the TEER values drop significantly. And that's more than what they would drop just, without doing anything here in green. And that, again, has been repeated many, many times. And so we can really consistently see that this is the case, which is also one of the things that we believe happens in inflamed intestine, because you have more permeability, you make it more permeable because of the inflammation that has happened. So just to show you quickly what we've done with this. So we have had the TNF, stimulation on these intestinal healthy organoids from dogs. And then we gave it DEXA, or we gave it DEXA and TNF. And what you can see again is that DEXA basically, alleviates this drop in TEER values, which is also something, of course, that we see clinically, because if we give, immunosuppressive doses of dexamethasone, the permeability is reversed, and basically you can stop the diarrhea. So that was kind of a nice confirmation of our assay, and, something that we can use as a control as well, if you wanted to, for example, test any compounds that you don't know whether they have anti-inflammatory activity. And you can say, if you give DEXA, it restores it. What happens if you give your compound? And then the next step, of course, is using organoids from diseased dogs, which I think is really going to be one of the things that we want to invest a bit more time and effort in the next few years, potentially. And what I want to show you here is that we had ileum organoids from a dog that was diagnosed with protein-losing enteropathy. This one on the left here is from a healthy dog, also an ileal organoid. And what you can see here is that the apical membrane on the inside here, right? So looking towards the inside of the organoid, you can see that it expresses sonulin here on the apical membrane. The difference to the organoid from the dog with PLE is that sonulin expression is now much, much stronger, and it's basically not just expressed apically, but also in between the cells. And what that means potentially is, when we want to translate it to what we see clinically, that you have much greater permeability in this organoid, and probably also in vivo. And so those are things that, of course, we need to work on a little bit more, get more characterization going. But these are things that potentially could be really important, because we might be able to reverse some of the effects that we see on the intestinal epithelial in these dogs, and maybe, move away a little bit from this maybe obsession with intestinal inflammation and how many T cells we see there or not, but maybe focus a bit more on what the disease process is in the intestine itself, in the epithelium. And just quickly to show you here as well, we have moved on from intestinal organoids and have just published last year that we actually have a lot of other epithelial tissues, and that also includes all endocrine tissues as well. So we're able to culture any type of endocrine as well as epithelial tissues from dogs, anything you're interested in looking at. And then for the last few minutes, I also want to show you how a disease model can be used in the context of intestinal, in this case, gastric intestinal organoids. So gastric cancer is a big dilemma still in human medicine. There's about 1 million new diagnoses per year globally. Almost all of them, about 800,000, die per year. It's a difficult disease, because a long time before you diagnose it, you have a lot of unspecific clinical signs, which means that by the time you diagnose it, it's usually very late. They are usually metastasized at the time of diagnosis, and they're very difficult to treat, because at that point, they're very aggressive as well. So at this point, a lot of times people will try chemotherapy as well as immunotherapy combination treatment. But so far, there's really no breakthroughs in making survival, really being much better than before. So the molecular pathogenesis in humans of gastric cancer seems to be through gastric inflammation. And then what happens is that you get a stepwise progression from chronic gastritis to atrophic gastritis, intestinal metaplasia, intestinal dysplasia, and then intestinal type carcinoma. And so this is really interesting because it seems to be that we have actually with the dogs that we recently diagnosed to have a higher prevalence of gastric cancer, such as the Belgian Shepherd dogs, to have sort of a similar progression of this carcinoma in the stomach. So treatment in humans is done with chemotherapy, like platinum compounds, radical gastrectomy, also HER2 antagonists, and immunotherapy. But even with all of that, the overall survival in metastatic gastric cancer is less than a year. So it's really not solved. And so my question was, can we help with our novel or new population of dogs that develop gastric cancer? Can we, get gastric cancer organoids from these dogs, then test novel drugs on these organoids from dogs as well as organoids from people before we take them into clinical trials in dogs and then into clinical trials in humans? So in dogs, as I said, there's this recent increase that's been recognized in the prevalence due to risk associations in Tervuren and Groenendals particularly, but also in other Scandinavian breeds. Again, just like in humans, it's diagnosed very late because of non-specific clinical signs, but the histopathology is very similar, and they also have these subtypes of intestinal and diffuse subtypes. And again, it's really difficult to treat because they're usually metastasized at the time of diagnosis. So I have this really nice collaboration going on with Thomas Spillman in Finland, who has a clinical trial going on, which is a prospective clinical trial where he sees Belgian Shepherd dogs about every few months, and he diagnoses them with either pre-neoplastic, like the metaplasia or dysplasia, or neoplastic lesions like gastric carcinoma. And he uses chromoendoscopy, which is quite a bit better at narrowband imaging actually as well during endoscopy, which is a lot better than the normal light endoscopy, where you can pick up these lesions a little bit better. And so it's easier for him to identify them and then also take biopsies from different regions. So far, what I can show you is that we had 12 dogs that had lesions spanning from healthy adjacent mucosa to metaplasia, dysplasia, and then gastric carcinoma. Some of them actually had several of them in the same dog, but most of them have predominantly either pre-neoplastic or neoplastic lesions. And so what we've been doing is basically culturing all of them. So we now have 12 organoid lines from dogs with either chronic gastritis, metaplasia, dysplasia, or gastric carcinoma. And what's interesting to see here is that they really look very different depending on the histological diagnosis. So this is from a dog with gastric metaplasia. This is from a dog with gastric dysplasia. You can see that the organoids really seem to be budding quite a bit more. And then this one is from a gastric carcinoma. And with this one, you can see that it basically tries to escape the little well that they're growing in. And this is usually for organoids a sign of invasiveness, and they could form metastasis in vivo. So obviously that would be really nice because we could use that for testing of drugs and also to see if they potentially work even in pre-neoplastic lesions so that it never actually progresses to gastric carcinoma. And that would obviously be a really nice way of testing these things for humans as well. So in summary, how can non-model organism, and I mean dogs in this case, with 3D organoids be used to help with current problems in preclinical drug testing? I think I've shown you that for toxicology and intestinal absorption, we can use healthy canine intestine. Although it's not perfect, it's probably still a lot more similar to the 2D cell lines that are being used. And then if we look at efficacy testing, organoids derived from diseased dogs with analogous diseases are obviously also something that we can look at. So here we have the model of intestinal inflammation, where we perform TNF stimulation of healthy dog organoids and then use compounds to see if we can counteract the IL-8 that's produced by the epithelium. We can also use organoids that are derived from chronic enteropathy dogs, like the ones that I've shown you from the PLE dog. And we can also use organoids from gastric cancer in dogs. So that's all I have. I just wanted to let you know that all of our data on the organoids from my lab are uploaded on the NIH depository that's called Integrated Canine Data Commons, which hopefully, will make it more accessible to everybody. And we're happy to share all of this because I think this methodology really has a lot of potential, and we want to make sure that people have access to it and can use it as much as possible. So in the end, I would like to thank everybody in my lab, because without them, I wouldn't have any of these data. My research scientist, Lina Martinez, my PhD students, Jia Peng, Somia Goddess, and Brian Melvin, the vet students that are in my lab, Lawson Brinkley and Sarah Towns, and the undergraduate student we have right now, Ethan Polsky. And please reach out at the email at UGA that I have there, and I'm happy to take any questions. And thank you so much for listening.
So we can thank Karin. She's not connected, I'm right? Do you see Karin? No, I don't think. Normally she's not connected. So maybe if you see, Amandine, a few questions on the chat, do not hesitate to hand your hand. Is there any question in the audience? We have a few minutes to discuss this aspect. Maybe I have two questions, but I don't know if the scientists in the room can answer this question, Karin being absent. The first one is, we haven't seen any bacteria, any crosstalk between the microbiota and the organoids. Do you think, the scientists in the room, do you think that it would be a good tool to study the interactions between the microbiota and the host? I don't know if Moez, Emmanuel, Cédric can discuss about that. I don't see Cédric, where are you?
Well, obviously
just you have the microphone, Amandine. Okay. Yeah.
Perfect. Thank you. So that's what we do actually. I will not be able to show much of these results later, but indeed it's relevant. It's the closest that you can kind of get currently in order to have a representative system that is obviously integrating the microbes, which are ubiquitous and which play important roles. We know very little about this, which is even more so important that this is pushed forward. That's a great resource. I was not aware of that really. So it's also a massive effort, and unfortunately also massive financial efforts, because this Matrigel stuff is super expensive. For anybody who has done this, they would probably attest to it. Yeah, it's just it's a great model, but it comes with its challenges.
Great. So, Emmanuel can add something. Yes.
Yes. To complete on that, I think it's also interesting if you consider testing drugs, because we know that many drugs, the microbiota interact with many drugs, and that it can be really key to get responder or non-responder, and there is more and more information on that. So I think that what would be, it's very good that FDA recognizes time of model. It's really one step ahead, but I think we should also consider, if we want to test drugs and drug efficacy in vitro model, we should really consider the possible interaction between the drugs and the microbiota. We know that sometimes it's just destroying the efficacy. Sometimes it's really key to have active treatment. And also, for example, in terms of interaction with the cell and gastrointestinal cells, it's also very important. Sometimes, due to modification of the microbiota, the drug can go through into the cell or not, or stay in the gut and be eliminated. So it's another dimension that I think we should really consider when we want to test the efficacy of drugs in vitro system. Yeah.
Yes, another question.
I was just wondering if we're also doing testing in other species that may be more, that are thought of as being a little bit more related to human cell lines, like pigs, for example, or not?
Do you have any comment on that, Emmanuel?
Do you want to comment?
Yeah, please go ahead.
Yeah. Can you repeat what is closer to human cells?
So I was just wondering if, in addition to dog organoids, we're looking at maybe some species that could be even closer to human physiology, like
okay.
pigs.
Yeah, yeah. I think there is also organoid development in pigs, but I can only stress what Cedric said. It's really a huge investment. So it's not, but it exists already, and there is more and more interest in that, of course.
And are you aware of any development or to make the organoids more affordable financially? It is very costly.
Yeah, let me just to be clear. Today we have several organoids from different species, from human. Excuse me. From human, pigs, dogs, okay, but the question, the Europe at least, they push in order to use organoids instead of animal models. But the question today, they are very costly, and we need several development, and so by the way, we need actions, so by the way, money in order to develop that. Lot of consortium also working on that. So I know, Emmanuel, that you're involved in several strategic commission in order to think about that. What is your point of view? And also from Cedric, I think that organoids today are good. People, they are talking about making them in contact with microbiota. But what about also to use them in another system like gut and chips, and what are really the outputs? What are really, it is a step forward, or we are still now facing technical problems that, by the way, we need more development works?
Well, I would say that you can do this nowadays. The models are quite representative. Of course, it depends on the question that you're asking. So you can develop them for certain things. So you will see later, I come from an institute which is interested in neurodegeneration, and there are several models, organoid models for neurodegenerative diseases, but also from other departments that work on cancer. So you can actually get human organoids. That's possible. But again, it's an investment. It's time consuming in order to get those cells, because you have to start with them. You make a contamination and you can throw your whole batch away. So that's all challenging. And importantly, the costs, I mean, the only thing really to get there is if somebody comes up with a cheaper way of growing them, or if companies decide to make it cheaper in terms of the consumables.
Yeah.
These are the two main ways you could approach this that I could think of right now. And yeah, like you said, for the gut on chip, I will briefly touch upon this in my presentation.
That's great. Yeah.
It's it builds on this Transwell setup. So and also Roxanne has the device with her. So it's actually a gut on chip device that we have developed, which you can look up. She also has a poster. Shameless plug, by the way, for her work. It's what I find really interesting about the IBD Day, so I am here the second time, is that to see that in animal research, the same challenges come up as in human research. So that's really interesting, and I don't really not know yet how to reconcile this, but I hope there are ways, because it's stupid to duplicate efforts.
Yeah. So we learn to better to do. Okay, please. Yeah.
Well, just I was wondering actually if dogs, like human organoids, another challenge that we will face is that there's really different between patients, and you can have from one patient a culture is growing and you have no problems to develop it. But from another patient with the same disease, it's going to be totally different, and you will face many challenges, and sometimes the culture will never grow. And I was wondering if in dogs we will have the same issue that between different kind of dogs who have the same difficulties and the same differences that will make a challenge just to test drugs, actually.
We'll explore that by next March in our lab. Thank you so much. We should stop here and move for the next session. Thank you so much.